Okay, there are two different types of drawings. Number one is called what? Detailed drawing, and number two is called what? Plan drawing. Okay, examiner can ask you to draw any type of drawing. Okay, so what is the meaning of detailed drawing? Detailed drawing is the normal drawing that you people know, are aware of it. So for the drawing, you are having 4 to 5 marks. In the detailed drawing, you need to show cells and all the visible structures. Okay. Now, what are the rules for drawing the detailed drawing? Number one, the line should be clear and continuous for the detailed drawing. Okay. Not sketchy. What does it mean? For example, you need to draw a clear continuous line. It shouldn't be like this, or a discontinuous line, a slight discontinuous line. So both of them are wrong. Only this line is correct. Okay. It should be drawn with a sharp pencil, no shading. For example, what does it mean? For example, if you are provided with the structure of leaf in which there is a shading on the leaf, on the midrib, plus it has a certain region which contain chlorophyll in it, and the outer region, it does not contain chlorophyll, or it is lighter in color. Okay, let's suppose the inner region is darker in color, because of course in your exam paper the diagram which is provided is not colored. It is uncolored diagram. So let's suppose the outer region is darker in color or white in color, and the inner region is lighter in color. Okay. And this is your stock, which is shown in the diagram. Okay. So now when you are going to draw the diagram, so how you are going to draw it? At first, you need to draw a large size diagram. And you cannot shade the diagram. If you will shade it, then you will lose 1 mark. So no shading. Okay. And you need to show these two distinct regions. How you are going to show it? You are going to show it with the help of a line. Is this clear? Okay. So shading is strictly not allowed. Previously, the diagram was not well proportionate. This shaded region was in the middle. Therefore, we have to show it in the same way. Okay. So shading is strictly not allowed. If you are going to shade this region, this is wrong. We don't need to shade it. So this is right. This part is wrong. Is this clear? Okay. And if for instance, if in this leaf veins are present, of course examiner is going to show the vein with the darker line. But how you are going to show the vein? Again, you need to show it with double line, but it shouldn't be darker. How many veins are visible in this diagram? Only two veins are visible, and their arrangement should be same. Okay. And let's suppose if the examiner asks you to label the diagram, then you need to label the diagram. So what is this part? This part is lamina. How you are going to label the diagram? You are going to label the diagram using ruled line. So no need to use the ruler, but you need to use a straight line, and that straight line should touch that part. For example, if the examiner asks you to label the midrib, how you are going to label the midrib? Let's suppose if you have labeled the midrib in this way, midrib, this is a wrong labeling. No mark will be rewarded. How you are going to label it? You need to label it by touching the midrib. Okay, so this is how we are going to label it. And what is this part? This is a leaf stalk. Okay, so for any drawing to draw, for any detailed drawing to draw, number one, there should be clear, clean lines and continuous, not sketchy, not sketchy, not discontinuous line, not discontinuous line, but a continuous line, and no shading, no shading, it will get wrong, and different parts of the specimen are in correct proportion. All of the structure on the specimen are included in the drawing. That is, all the veins should be shown. If the diagram is divided into two distinct regions, then you need to show these two distinct regions with the help of lines. Okay, and as in this diagram, four veins are clearly visible. So you need to draw four veins, but no, avoid shading it. Otherwise, you are going to lose that mark. Now, label lines are drawn with a ruler, no arrowheads. Sometimes students label the diagram by using arrowhead on the label line. It will get wrong. Why is that? Because what arrowhead indicates? Arrowhead is indicating direction. So this is not showing any direction. So always label it using straight line. Is this clear? So no need to label using arrowhead. Okay, so this is a midrib. Then labels do not cover any part of the actual drawing. What does it mean? Sometimes student label within the diagram. How they are going to label vein? They label vein sometime within the diagram. So it will get wrong. You need to label the diagram outside the diagram, not within the diagram. Is this clear? Do not use ruler or compass to draw. Sometimes, for example, if a circle is drawn, then students may use a compass or ruler in order to draw the diagram. So avoid using it because a biological drawing should be freehand. Okay. Read the question carefully to see if any specific segment is required. Do not draw. What does it mean? I'm going to tell you just after a while. Okay. Do not draw the surrounding box of the image. What does it mean? I'm going to explain it later in a short while. Now, what is a mnemonic for drawing? It is sold. S means more than 80% of the available space should be occupied and correct proportion of the parts. Clear continuous outlines, no shading, labeling if asked, details visible on the diagram. That's how you are going to get 4 marks or 5 marks. Okay. Now we'll come back to it. This part later. Now, now plan drawing. What is the meaning of plan drawing? As there are two different types of drawing: detailed drawing and plan drawing. Now, what is the meaning of plan drawing? Plan drawing means you need to show the distribution of tissues in a specimen. No need to draw individual cells. Okay. Now, what does it mean? So let's look at the plan drawing first, then we will see the detailed drawing. For example, the examiner provided you with this diagram and asked you to draw the plan drawing. And let's suppose if the examiner doesn't mention the name plan drawing, and if the examiner asks you to draw this diagram and avoid drawing individual cells and label certain parts. So how you are going to draw it? You need to show, as this is an epidermis, this is an epidermis. Okay, so this outer layer is an epidermis. Okay, so as the epidermal cells are like this, are barrel shaped cells. So as it is double walled, so how we have to show it, these epidermis, if we have to draw it, how we are going to show it, the outer layer of these of this tissue, how we are going to show it, or of this structure, how we are going to show it, we need to show it with double line. So first line and then the second line. Okay. So, okay, and you should draw it with the clear, very sharp pencil, not with thick pencil. Okay, and that pencil should be lighter. Okay, not very dark. Okay, and then so this is an epidermis, and then you need to show these holes. How we are going to show these holes? 1, 2, 3, 4, 5, 6, 7. We need to show approximately 6 or 7 holes on this diagram, and then over here within this structure there lies vesicular bundle. Of course, a clear diagram will be provided by the examiner, not this vague diagram. So if the examiner is going to provide you, because this is the TS of the root, we know that in the TS of the root, if the examiner asks you to label the location of xylem vessel, so we know that the vesicular bundle of the root, it looks like this. You need to show again with the double line because it contains endodermis. Again, endodermis are again barrel shaped cell. So in order to show its thickness, you need to show it with double line. And then there lies, these are the phloem vessels. And within these phloem vessels, there are star shaped xylem. So this is star shaped xylem. So this one is xylem and this one is phloem vessel. Clear? So this is called what? Plan drawing. Another plan drawing, for example, examiner provide you with this dicot stem. Again, how you are going to show the epidermis. So this is not a very good diagram. How you are going to show the epidermis, we need to show it with, draw it with a lighter pencil. Okay. So how we are going to show, draw the circle, do not draw with compass. Every biological drawing should be freehand. I will lose 1 mark for this discontinuity. Okay. There shouldn't be any discontinuity in the drawing. Now don't judge me because you cannot penalize me. Okay. And then we need to draw it with double line. My line can be discontinuous, but yours cannot. Okay. I'm drawing discontinuous line. I will lose 1 mark, but it shouldn't be discontinuous. Okay. Now, after drawing it, you need to draw these vesicular bundles. So how many vesicular bundles are shown? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. So you can draw 10. Okay. But not less than 10. Okay. So draw these vesicular bundles. Of course, they should be a slightly bigger in size. When we are drawing in our journal, it will be easier for to draw in journal rather than drawing on the screen. They are in fact like this, not, they are thicker from the edges and towards the center they are thinner. Okay, so examiner will look for these vascular bundles. 1, 2, 3, 4, 5, 6, 7, 8. You can erase it and draw it again. Okay, so there are 3 distinct regions in these vascular bundles. You just need to show it with the lines. Do not draw any cell. In case of plan drawing, we don't need to draw individual cell. Okay, is this clear? Clear? So now you have to complete the entire diagram. Is this clear?
Yes, miss.
Okay, so this one, let's suppose examiner asks you to label the vascular bundle or label the xylem and phloem in the vascular bundles. How you are going to label it? This part is what? Is phloem. And what is this region? This region is xylem. And what is this? This is epidermis. Is this clear? No.
Miss,
You
like
need to draw
won't we
all these
draw the
vascular
cell
bundles correctly.
membrane too?
Cell membrane? Why cell membrane? Is
Yeah.
this a cell? This is not a cell. This is a TS of stem.
Ah,
TS means transverse section of stem. Okay, this is not a cell. Okay. Now draw these. You need to draw these vascular bundles correctly because these are incorrect, because these vascular bundles are, as per this diagram, are thicker from the, are thicker at the edges. And thinner at the bottom. Okay, so you need to draw it in this way. Clear? Clear how to draw plant diagram? Okay.
Yes.
Hello. Clear? Now, no. These are the diagrams. For example, if this diagram is provided by the examiner, and if the examiner asks you to make a large drawing, sometimes examiner can ask you to make a large drawing, sometimes examiner can ask you make a drawing of same size as shown in the, in figure 2.1 or whatever the figure it is. Okay, so if the examiner asks you to make a drawing of same size as shown in figure 2.1, so it should be of the same size. Is this clear? But if the examiner asks you to make a large drawing, so what does it mean? What's the meaning of making a large drawing? Large drawing means you need to occupy 80% of the space provided. What's the meaning of this space? Space means, for example, if the question is written over here, and if this much space is provided and the marks are given on the bottom right corner. So this is the image, this is the space that is provided by the examiner. So you need to occupy more than 50, more than 80% of the space. Is this clear? Now, rule number 1. Orientation of the diagram should be similar. Now, what does it mean, orientation of the diagram? If the diagram is showing in this direction, if the image is shown in this direction, then you need to draw the diagram in this direction. You cannot draw in this direction. Is this clear? Otherwise, you will lose 1 mark. Okay? Okay? Now, next, the diagram should be drawn with the clear continuous line. Now this fruit, this rosehip fruit is more wider as compared to its length. So you need to show it in the diagram as well. Okay. that the rose hip fruit should be more in the width as compared to its height. Okay, then you need to draw this fruit stalk and then these leafy structure that I'm going to ask you. What is the meaning of this? What are these leafy structure? Okay, now when the examiner asks you to make a large drawing of the rose hip shown in the box in figure 2.1, label a sepal on your drawing. So how you are going to draw it? Do we need to draw these leaves as well? Do we need to draw these leaves as well? Yes, because the examiner asked us to draw the rose hip shown in the box in figure 2.1. So do we need to draw this box? This box. Examiner is talking about this box, not this larger box. Examiner is talking about this box. Okay. So do we need to draw these leaves as well? Hello? No, we don't need to draw the leaves because the examiner asked us to draw the rose hip only. Okay, so we need to draw enlarged diagram of the rose hip only. Okay, now, so this is the first part. What are the rules of our diagram? Size more than 80% of the space occupied and correct proportion of the parts. So what are the parts of this diagram? The parts of this diagram is this fruit stalk, this fruit, and this leafy structure. So how you need to draw it? At first, orientation of the diagram should be same. Number one. Number two, the diagram should be well proportionate. That is the size of this stalk and then the size of the fruit and then this leafy structure. It should be proportionate, well proportionate as shown in the figure 2.1. So what do you think? What do you think about this diagram? What do you think about this diagram? Will it get 4 marks, 5 marks, or what? What's the meaning of details of the diagram? Details of the diagram means you need to draw as many number of sepals, or you can draw only one less sepals as shown in the diagram. Let's suppose if in this diagram 5 sepals are shown, so you can draw 4 sepals, but not less than 4. Okay, and this sepal should be delimited. Okay, it should be delimited. You cannot draw it in this way. That is, you have drawn this fruit. Let's suppose if you have drawn this rosehip fruit and then if you draw sepal in this way, then 1 mark will be deducted. Why 1 mark will be deducted? Because you haven't delimited this fruit or you haven't delimited this sepal. Okay, so always delimited. Okay, and then you need to draw this fruit stalk as well, and it should be well proportionate, so it shouldn't be this long. Okay, it should be well proportionate. Is this clear? Okay, and line should be clear and continuous. As these are the sketchy lines, so -1, it should be -1. Is this clear? Now, examiner asked you label a sepal on your drawing. So where is the sepal on your drawing? Do you know where is the sepal? Where the sepal is located in this drawing? This is a sepal. So how you are going to label it? You are going to label it in this way with a ruled labeled line. Is this clear? So that's how you will get 4 marks out of 5. Why 4 marks? Because of this discontinuity. Is this clear? Is this fruit wider than its length? Yes, 1 mark. Are the line clear and continuous and no shading? No, you will lose 1 mark. Details of the diagram are there? Yes. Labeling is correct? Yes. Are there delimitation? Yes. Is this clear? Okay. And more than 50% of the space is occupied? Yes. Okay. So that's how you will get 4 marks. Why 4 marks? Because of this discontinuity. Is this clear? Now look at the second diagram. The examiner provided you with the second diagram. It appears in June 2025 of 5090 paper, not 0610. Okay, paper 62. Paper 62 means paper 42 because in 5090 we have paper 4. We call it paper 4 rather than paper 6. Both of them are almost same. Okay, so no need to worry about it. If it is 5090 0610, it don't make any difference. Okay. Now figure 2.1 is a photo micrograph of a section of a celery plant. So this is a figure. Okay. So what do you think? How we need to draw it? How we should draw these vascular bundle? What do you think? Do we need to draw it in this way?
Yes.
In this way? Do we need to draw
No.
vascular bundles like this? Do we need to shade
No.
it? Shade
No.
these vascular bundles? Hmm?
No.
No, we don't need to shade it. Otherwise, we are going to lose 1 mark. Now, how should we show this lower line? Should we draw it with double line or with single line is fine? Just
Double
look at
line.
the diagram closely. Very good. Why do we need to draw it with double line? Because it is a thicker line. So in order to show a thicker line, we need to draw it with double line. So the lower line, we need to draw it with double line, and it should be like this. It should be drawn with the double line, and it should be clear and continuous. Let's suppose your drawing is very poor. Always draw an enlarged diagram, you will get 1 mark. Don't shade, and always draw a continuous clear line. You will get 2 marks. Okay, if your diagram, let's suppose if your diagram is very poor, at least try to do these two parts. Okay, make a large enlarged diagram if the examiner asks you to make a large drawing. If the examiner asks you to draw a diagram which is of similar size, then no need to enlarge it. Okay, just measure it and draw the same size diagram. Okay, now and then you need to draw these scalloped edges. These are the scalloped edges. Examiner will look for it. Whether you have drawn these scalloped edges with double line or not. If you haven't, you will lose 1 mark. If the details on the edges are shown, if you don't draw these details, you are going to lose 1 mark. Okay, so these are the detailing. Okay, so this is how we draw a detailed diagram. Is this clear? Because this is not a planned diagram. So we have to complete it, and then we need to draw these vascular bundles without shading it. These are the vascular bundles. We don't need to shade it. Is this clear? So I'm going to show you a 5-mark diagram and 4-mark diagram. Wait. So this is a 5 marks diagram. This is a perfect diagram. Scalloped edges are shown. These detailing on the edges are shown. The vascular bundles are drawn with the without any shading. Line are very clear and continuous. Okay, and it is drawn with the double line. All the lines are shown with double line because all um, uh, um, the external lines are drawn with thicker lines. That's why we need to draw it with double lines. Okay. Now this is a 4-mark diagram. Why 4-mark diagram? Because the double lines are not shown. Vascular bundles are drawn, but they are drawn with the shading. That's why it will get wrong. BOD means benefit of doubt, whether the diagram is of 90 millimeter size or not. The examiner has given the benefit of doubt for it. No shading, the person has shaded it, therefore that person has used his marks. Now no clear, clean and continuous line, so that person will get 1 mark. Concave bottom outer edge plus scalloped upper edge, so we'll get 1 mark. Vascular bundles are drawn. Okay,